All right, good afternoon. They say spring is finally here, but it's not. The weather is crazy recently, right? You must find yourself making decisions constantly whether to take on the actual layer, down to the micro scale. The cells in your body are constantly making that decision either. The cell adapts to the new environment, such as a colder place, by changing its related gene expression. For example, when the room is hot, the cell might want to take off the actual layer, and thus the gene expression is low. Thus, to understand how cell adapts, it's important to understand how gene regulation works. So as in the previous biology talks that you just heard today, I have to show some cartoons to illustrate how that works, right? But as in my title suggests, I'll definitely replace it eventually with equations, which are my favorite. So please focus on the figure on the left. As shown, you can think of the gene regulation as passengers looking for only one VIP seat in the train. So how does that work? The RNA polymerase and repressors, as passengers, are walking randomly on the train of this DNA sequence. They are looking for the special VIP seat in the top part, which is the white sequence, is their binding site that they are competing for. So once they find the binding site, which is the VIP seat, the RNA polymerase would initiate the gene expression, which you can think of to put out more layers. And the repressor, on the other hand, would inhibit the gene expression, which means to take off another layer. Thus, the cell's decision of whether to express this gene depends on which protein of the repressor or the RNA polymerase gets to occupy that binding site, which is the VIP seat on this whole train. But how can we predict the gene expression level from this cartoon? As physicists, we are not satisfied. We want to capture everything in a simple equation. The idea turns out to be really, really simple. It's all about counting. So to find the level of gene expression, it's the same of asking the probability of either kind of the regulatory proteins getting to occupy that special seat. So we can simply count the configurations that the VIP seat is taken by either the repressor or the RNA polymerase and divide that by the total number of possible configurations. From this, we can predict quantitatively how the gene expression is controlled by its regulatory proteins. So eventually the cell's decision process can be all captured by this single glorious equation on the right. But you may ask, how does this equation help us understand how cell adapts in different temperatures? Well, it turns out that temperature may influence the activity of repressor. And we can include this effect in this equation as well. So this is exactly what I did in my research. And I showed how exactly the gene expresses in different temperatures. So going back to the beginning of my talk, now I know what temperature exactly the cell feels cold or hot, and how it responds to the changing temperature. So this can have potential applications for drug design that's sensible to our body temperature. And this is what I want you to take away from this talk. Equations are not just numbers. They are a superpower that helps us to understand nature better. Thank you.